And I'll start with. So again, good afternoon. So we are 5 minutes already. And lima paghihapon mo. Anyways, I'll just have screenshot first before ta maka-start. And for those who will miss this class, naka ano man noon no, naka record. So today is 22. Ano? Okay, so last time I guess we stopped here no on still we are on prelim lesson 1 component ng foods. So last time we generally we identify the different, as you can see in our outline here, we mentioned about the different components ng mga food and then how to analyze them. So we stop on this analysis and characterization of food. So what is the importance of this one is that we will know no na the different technologies in identifying the different contents. Now, for example, itong high performance liquid chromatography. No, it is for determination of certain vitamins. So, if we are going to look on, have you checked na on perhaps on YouTube ba? High performance liquid liquid ba to? Chromatography. Yeah, chromatography. High performance. Nawala na nung ko. Ay, sa YouTube ba ko nag-search? Liquid chromatography. So, if we, have you checked in sa videos, no? Have you checked? Wala pa. So, or in short, this is HPLC. So, madami tayong pwede tingnan, no? This is about yung kaniyang principle behind. So this is what it looks like, no? And you will learn the different principle behind this.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a non-polar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Okay, so that's it, no? So we'll go back here. So that's HPLC, in short, high performance liquid chromatography. So those are one of just the few na mga analyzer, no, equipment that we have. We also have gas liquid na chromatography and its kind. So if you are going to search for gas liquid, although dili na naka na siya, this looks like this, no? Sa mga, like when you measure ethylene naman yan, no? See, so this is an example of the gas liquid chromatography, or GLC, no? Yan. So for mga gases, no, like one of example yung usually is ethylene para sa ripening ng mga ng mga ating fruits. So anyways, so those are the different equipment used for measuring the content of our food, no? And we also have for proteins naman, we also have the NIRS, or the near infrared reflectance spectroscopy. So let's try to see NIRS. Sorry, NIRS. Okay, so ano itong NIRS, or near infrared? Let me check. So it looks like this one. Ito, no? Ito. So lahat ay merong parang box na equipment, and then merong monitor to show kung ano yung meron, no? So ayan. And going back again sa spectroscopy, so different siya sa chromatography. So this is more on, although general siya na element ang HPLC or chromatography, but sa spectroscopy, iba yung ginamit na kanyang principle. Like you use the infrared reflection. Kasi yung chromatography, you use the absorbance. Yes. So ayun. So and we also have nuclear magnetic resonance spectroscopy, which is more na mas high-tech pa jud talaga, no? So in summary, so the thing that we need to remember is that water is one of the major component ng ating mga different food, no, and including the animals. And this contains carbohydrates, proteins, vitamins, minerals, lipids, and there are analytical techniques to use them. I mean to measure them, no? This include chemical and gravimetric determination, and even other modern analytical techniques. So for example, starch is determined by polarimetry. Yan, meron din tayong fibrous constituents, GLC, and minerals using spectroscopy, and GLC again for fatty acids, certain vitamins, and near-infrared reflectance spectroscopy. So, madami. So you just have to, those are the references, and you just have to answer only few questions. This were the video we had last time for those who miss it. And again, I am going to include also itong recorded na ito. So do you have any questions or clarification before we proceed sa ating next na next na lesson? Do you have any question? So we are now on carbohydrates. Wait lang. Humana ba tag carbohydrates? Wait lang. Wala pa ata. Please correct me kasi maybe I just forget. Skip. So, did we start with carbohydrates? Not yet. Wala pa ba? Okay, anyway, so check ko muna ulit yung ating.
Not yet pa po.
Wala pa, no? Ay, thank you, Shine. So, these are just introductions sa lahat, no? And pangit kay ko. So, introduction lang yan sa lahat ng ating prelim pa lang. But anyways, let's just take time. Kasi, wala pa po na ako na-upload lahat, no? March 22. Okay. So now we are on carbohydrates. So in this lesson, we are going to talk about the different classification ng carbohydrates, no? And whether they are monosaccharides and its derivatives, or whether they are oligosaccharides, polysaccharides, and lignin. So last time, I was only very familiar of disaccharides and monosaccharides. No, when we say mono, of course, isa lang, no, single sugar, I mean sucrose for saccharides and disaccharide polysaccharides or ano. But we also have a lot, no, even lignin. And then in general, lignin was not actually sa inyong quiz na siya, no. So I leave that to you. So anyways, yung ating general carbohydrates na groups are actually mga aldehydes, ketones, alcohols, acids, and mga derivatives. So lahat ng mga compounds na merong carbon, hydrogen, and oxygen, or we have the CH2O, are actually carbohydrates which contain phosphorus, nitrogen, sulfur, and other. Like example, the deoxyribose, the C5H10O4. So actually, there's no need to actually dig deeper into this kasi this is very biochemistry. And huma naman mag-biochemistry, no? Third year? Mana mo? Memorize na ninyo ang mga chemical formula sa carbohydrates, proteins, so kabalo mo unsay alcohol, kabalo mo unsay kuan, anything, no, na yes, kasi na nag-chat. Wala, ha? Wala pa mo, Ken? Wala mo yung biochem sa inyong
Chemistry lang, ma'am. Walay biochemistry.
So for chemistry, general biochem siya, no? Okay. So so we have here classification ng carbohydrates. How many slides do we have? So we have 16 slides. So supposedly mubura ni. And so classification, we have simplest sugars. Ito yung mga monosaccharides. If you remember, diba? Of course, na-remember nyo naman to. And simple kasi mono, isa lang siya, no? And these are divided as trioses, tetroses, pentoses, and hexoses, heptoses. Depende yon sa carbon present sa molecule. So you can see here, carbon is 7 in heptosis and hexosis C6. Of course, triosis is from the word trio, 3, no? And pento, 4. And trio 3, tetro pala 4, and pento, diba? Pentagon is 5, pento 5, and hexo 6, and hepto 7, no? And unsa na sunod ana? So those are monosaccharides, and we also have pag ma-link na siya, this can be di, tri, tetra, poly, no? Pag i-link na nimo ni mga monosaccharides, kaya magiging ano na siya, disaccharides, tri, tetra, and so on, no? So yung sugar is carbohydrates, hi Paulo, asa ka Paulo, containing fewer than 10 monosaccharides. Okay. Fewer than 10 monosaccharide residual oligosaccharides is from the Greek word oligos, which means few lang siya, no? And these are used to include lahat ng sugar na monosaccharides. So 10 pag sinabing sugar, mga 10 monosaccharides lang. So polysaccharides are glycans or polymers ng monosaccharides. So daghanan na siya. Makita niyo yung substructure, no? And classified as homoglycans, or single type lang siya ng monosaccharides, or heteroglycans, which on hydrolysis yield mixture ng ito. So ito ang mga examples ng ating sugars. So classification ng carbohydrates in figure. So for sugars, we have mono siya or oligo, no? Sa mono, meron tayong, yun nga, trio, tetro, pento, hexo, hepto. And these are again classified as glyceraldehyde, dihydroxyacetone, erythrose, and so on, no? Pag pentose or limang, pag tetrose, erythrose niyan. Pentose is, no? Arabinose, xylose, and ribose, cellulose, ribulose, glucose. So ito na, no? We have glucose or the table sugar. Galactose is the galactose. Galactose is the combination. No, hindi pa. And mannose is also, ano nga yun? Fructose, of course, for fruit sugar. And heptose, sorry, I forgot. Pseudoheptulose, no? Example. And for disaccharides, of course, sucrose. I don't know, glucose pala is blood sugar. And fructose, fruit sugar. So those are hexoses. And for disaccharides, under oligosaccharides, sucrose, yun yung table sugar, lactose, of course, milk sugar, maltose, malt sugar, and cellubiose yung sa cellulose. So those are disaccharides. And under trisaccharides, we have raffinose, ketose, and tetrasaccharides, stachyose. No. And for polysaccharides, we have homoglycans. Under this, arabinans, silans, glucans. And under glucans, we have starch, dextrins, glycogen, cellulose, and callose. And of course, you have heard all these words already. No? Nakadungog na mo, ane? Jonah, Aliyah, kadungog na mo, ane nga mga terms before?
Glucose.
Ha? Yes. Glucose lang imo na dungog.
Yes, ma'am. Gamay lang.
Okay. Anyways. So, that's all right. So, kasi these are like, for example, we can see starch for almost all of our plants, diba? And dextrin may be siguro sa dextrose na dunggan niyo na, no? Glycogen, sa mga vitamins, cellulose, of course, and callose, no? And under homoglycans, fructans, galactans, mannans, and glucosamines, inulin, leavan, no? And we also have non-sugars, no? Kasi these are classified as sugars and non-sugars. So, lahat ng polysaccharides are non-sugars, meaning parang fiber lang siya, no? They are very complex carbohydrates. So, polysaccharides either be homoglycans, yung na-mention na natin, heteroglycans. These are pectic substances. So, remember, pectic substances are in the middle lamella of fruits, no? Or tissues ng plants and animals, ng plants pala. Especially sa fruits, no? Kasi yun yung magsusoften pag mahinog. And we have hemicelluloses, yung mga fiber, exudate gums, kung like chewing gum, no? Yun yung ating mga chewing gum, acidic mucilages, and hyaluronic acid, and chondroitin. So those all have each mga uses and functions. And for complex carbohydrates naman, ito yung mga glycolipids and glycoproteins. So I hope it's very clear kasi clear naman dito. Kaya I really choose this very ano siya, heavy and bulky na na book, but it's really very informative. No, madami lang talagang babasahin. But just take your time. So, kanina sa ating analysis ng food, tanaw, 10 minutes na. So, kanina sa analysis ng food, we have seen na lahat ng component ng food ay identified using molecular weight, no? Yung kung kung kapareho niya kabigat pag analyze, so it means it might be carbohydrates and it might be a very specific kind or type of carbohydrates, yung mga ganon. So yung molecular weight therefore ng mga polysaccharides will vary about 8,000 in some and as high as 100 million gani, no, sa mga amylopectin. So ganun ka different yung mga components in terms of carbohydrates and sugars. So and they also are being affected by action ng mga enzymes or acids. So ito yun yung makakapag-break lang, no? And so these are the monosaccharides, their structure. So alam niyo naman kung ano yung mga monosaccharides, di ba? We have fructose, glucose, no? So these are the glucose, may letter pa nga, no? And what are their properties? So meron silang active aldehyde or ketone grouping. And these are reducing substances. So as I have said, there are only important points to this. So pentoses are one of the most important group in simple sugars, the arabinose, xylose, and so on. And these are the different pentoses, no? And meron silang arabinans, yung arabinose, and which is a component ng hemicelluloses. So if you remember the word hemicelluloses, yun yung mga nakikita natin sa parts ng plants, no? Sa silage, sa ating animals, no? So usually they are found in large quantities sa mga herbage, no? And yeah, ito yung mga kanyang structure. Okay, so the hexoses. So hexoses are the glucose and fructose, and they are very natural occurring na mga hexose sugars. And they also occur yung mannose and galactose sa plants, and also polymerize as mannans and galactans. So glucose is a great sugar or dextrose, and they are, if they are free, makombine sila in form, and they can, this sugar occurs sa plants, sa fruits, sa honey, sa blood, sa lymph, and cerebrospinal fluid natin, no, or animals, and are major component ng mga oligosaccharides, polysaccharides, and glucosides. So etong mga monosaccharides na ito or simple sugars are actually part na din na mga complex carbohydrates. So mo man na siya magsumpay-sumpay lang if you remember, no, our chemistry or biochemistry whatsoever. So we have also the fructose, no, or a fruit sugar. They are in the green leaves, fruits, honey, and so on, no. We also have the mannose. They don't occur free in nature, but they are polymerized as mannans sa glycoproteins. So they are distributed also sa mga yeast. So yeast is the one that we are using in bread making, no? These are actually fungi and molds and also bacteria. So meron yan silang mga mannose. And galactose, they are also free in nature, except for fermentation, and they are constituent ng disaccharide lactose sa milk. So that's why galactose, no? Meron din sila sa anthocyanin pigments, sa galactolipids, sa mga gums, and mucilages like for animals, no? So those are the hexoses or yung 6 carbons, no? And the heptoses or the 7 carbons, it looks like this. And these are monosaccharide containing 7 carbons, as I have said. So ano yung mga derivatives natin? So we still have 5 minutes. Phosphoric acids, esters. So ito yung, ang role nito is very important sa metabolic reaction ng ating mga living organism. So usually, they form from glucose and nag-esterification siya. Okay, and these are the glucose 1-phosphate or 6-phosphates. So those are next is the amino acids, no, amino sugars. So these are hydroxyl group, no, with carbon atom 2 and aldohexose na merong amino group. So when we say amino, they are nitrogen, hydrogen, no, NH2. So ito yung glucosamine and galactosamine. So or amino sugar. So malalaman mo siyang amino sugar siya kasi meron siyang again NH2. So deoxide sugar siya kasi meron siyang na replacement by hydroxyl group by hydrogen yields a deoxide sugar. So ito ata siya, no? At meron siyang ribose, deoxyribose, which is a component ng deoxyribonucleic acid, yung ating DNA, kaya deoxide sugar siya, no? And derivatives nito ay hexose, galactose, mannose, or fructose and rhamnose, no? Or heteropolysaccharides. So, mabilis lang tayo kasi 3 minutes na lang. And we have sugar acids and sugar alcohol. So, ito yung mga aldronic acids, aldaric acids, and uronic acids. And these are important kasi these are still part, no, sa mga bigger complex sugars. So, the sugar alcohols are the fructose, mannitol, and mannose. And the glycosides also are one of the derivatives na meron siyang attached na carbon 1 atom glucose. So, makikita natin yan sa next slide sa ating figure. And oligosaccharides and polysaccharides are also our glycosides na as seen here. So ito yung ating mga naturally occurring cyanogenetic glycosides. Yung nakikita natin sa product acetone, no? Sa linseed, java beans, cassava, and ito seeds ng wild veg, almonds, leaves ng sorghum, and clover, no? Ito yung mga substances. So I guess we will proceed with oligosaccharides or disaccharides by next meeting, no? Page 23. So do you have any questions or clarification? So, inana lang takadali, no? Kasi limited lang atong time. So, any questions or clarification? Wala. So again, how many classification tayo ng carbohydrates? Christine? Nawala bigla si Christine. Bitaw, joke ra. How about Albert, no? Or Aliyah? If you can speak, no? The better. Or Paulo? Ang dili maka-answer, pakantahon na ako sunod. Hindi na mo ma-attend. No? Joke lang. So, unsa?
6 or
We have
buok ang
6 classification ng carbohydrates. 6 ba yon?
5, ma'am. 5.
Okay, so if we classify them, no, there were into group 2, there are more than actually 6 or ano, because we root from 2 major classification, sugar and non-sugar, and then we have monosaccharides, and then we have oligosaccharides and polysaccharides, and then meron pa tayong lignin. So you can say just 2 major groups or 1, 2, 3, 4 or 5 major groups.